Sweet. Okay. Well, thanks, Lyle, for the introduction.
Wait, there's an echo.
I think if they turn off the echo. Good now? Cool. Sweet. Okay, well, I really appreciate the opportunity to be here and talk to you today about the method that we've been developing for characterizing structure in large-scale spike recordings.
Nope, never mind.
So, as you all know, neurons transmit information through action potentials, which are discrete events that many of you have probably heard out loud if you've ever played the recording from an electrode directly through a speaker. And the spikes of an individual neuron sound like distinct clicks if you do that, but new technologies mean we can record the activity of hundreds or even thousands of cells at the same time. And if you tried to play that through a speaker, the result would be like listening to thousands of telegraphs clicking all at once. Now, if the telegraphs in my analogy were all sending messages independently, then we already have the mathematical tools we need to try to recover those messages. But on the other hand, you could imagine that some of those telegraphs might be coordinating to send messages, which would then be interwoven across the clicks of those telegraphs and over time. And we don't yet have the mathematical tools to recover the resulting complex and overlapping structure in those discrete events. So today I'm going to introduce to you the details of a new mathematical transform that we've been developing to handle this challenge that can detect structure in large-scale recordings and relate that structure to behavior. So in more detail, I'll start by talking a bit about the background and other methods for analyzing these large-scale recordings, many of which focus on firing rates, but there are also some computational algorithms for spike recordings at this scale as well, and some mathematical approaches for spike trains. I'll then go through the big picture of our transform to sort of show you how we go from a raster plot to the output of the transform where we can start making links to behavior. And then I'll go into some depth about the key mathematical details that make this transform possible. So we'll get into it later, but the main challenge is comparing spike trains or spike patterns across many neurons of different sizes, where different neurons can spike different numbers of times across trials, and you sort of have to handle that. And then at the end, if we have time, I'll show you some initial applications to experimental data. All right, so neural pixel recordings at the scale of hundreds of cells are becoming routine, and there have been a number of methods designed to handle these large-scale recordings. Common approaches like population decoding or state space analyses require first temporally smoothing the spiking data. And from there, there's a variety of approaches that can be applied to study these continuous neural dynamics. These techniques are extremely useful for distinguishing between experimental conditions. So for example, comparing neural responses during two different movements in a motor task or something. And for that reason, they've proven to be very powerful tools for decoding in BCI applications and things like that. But these methods are also designed based off of an assumption that spikes are stochastic samples of a true underlying rate that varies meaningfully over time to encode information. And as I said, there's a lot of robust methods that have been developed to test this hypothesis in large-scale recordings. And they've proven to be really useful. This is a nice review of some of them. But there's also an alternate hypothesis, which is that meaningful coordination could exist in the spikes across neurons at the level of individual trials. And there are fewer systematic methods that are available to address that possibility at the scale of these large recordings. So there are a number of computational algorithms that have been developed to address the possibility of coordination across large populations, often still in terms of the smoothed neural activity. So just to note a couple of examples, this adaptation of matrix factorization can find sequential structure in firing rates. There's a few different approaches for tensor component analysis, which looks for covariant activity across neurons, time, and trials, so you don't have to flatten along one of those dimensions to make your comparisons. And this is a kind of really interesting new one called raster map, which is a visualization tool that can help you cluster neurons that have similar responses together. So this one is applied to calcium signals. And those types of tools are really useful for exploring and visualizing these large-scale data sets, but they often require a number of parameters, and they're still operating on smooth spiking data rather than directly on the discrete spikes. Now, on the flip side, there's mathematical approaches that are designed for those patterns of discrete spikes. And those methods operate directly on your recorded spikes without smoothing, but they tend to be limited in scale, either in terms of the number of neurons that you can include or the number of spiking events per neuron. So for example, there's approaches for making pairwise comparisons between spike trains, like the Victor-Pepera distance, which is defined algorithmically by counting up the cost of transforming one of the spike trains into the other. And now that algorithmic process defines a clear distance measure for comparing single neuron spike trains. And of course, it's not the only spike train distance measure. So for example, these are some other approaches for making these pairwise comparisons, and they don't need an algorithm to define the distance. Instead, you can use measures like the inner product for comparing these spike trains, and they accomplish that by convolving with a Gaussian kernel in order to do that.
So that's the same who that just gave us a,
Exactly.
brief history of, he was here and he worked on those algorithms when he was here. We did a lot of work on looking for synchrony among spike trains, clustering, using clustering.
Yeah, and the one I noted here, actually, it has a really great like part about it in the difficulty of applying some of the like clustering approaches directly to a binary spike train, if you think about it in terms of zeros and ones, and why looking at it in terms of its similarity to other trials is really useful for like reducing that dimensionality. And that's sort of a like, you'll see later that we sort of go to a similarity space as well for that. But yeah, so these pairwise approaches, they can be hard to extend to large populations. And there are some numerical approaches that have been developed to take on that challenge. So this one, for example, extends the Victor-Pepera distance to like small populations of cells by adding cell relabeling as an additional parameter to the algorithm. And this one can go to large populations by making pairwise comparisons and sort of sorting the neurons based off of neurons that have similar types of responses. So in that way, it's kind of like the raster map one that I showed you before, but this time operating like directly on the discrete spikes. But even with those approaches, it can still be really difficult to analyze coordinated patterns across large populations. So one common approach is to reduce the complexity of the problem and instead consider only a single spiking event per cell. So often looking at patterns of first spikes or spike bursts. And if you do that, it lets you write out the population spike pattern as a vector of spike times. So here, each component in the vector corresponds to the spike time of one neuron. And from there, it's really easy to compare those vectors across trials and do things like find structure in the population by sorting those events in time to look at sequences. Now, that type of approach has led to some really interesting results. So for example, this work on first spike times in retinal ganglion cells during natural vision, or this one uses sequences of spikes in visual cortex to decode natural scenes. And they're making this simplification to a single spiking event per cell so that you can think about the spike pattern as a vector. And the reason that vector is useful is because it's a mathematical object that lives in a space where you can define a distance without needing to rely on a numerical algorithm. However, that type of approach is really dramatically simplifying the spike pattern that's being studied. And it's making assumptions about which of the spikes might be involved in relevant coordination. So for example, the first one or one that's part of the peak firing rate. So to understand why the mathematical approaches have been limited to those more simple cases, we're just going to take a quick look at a toy example of detecting a pattern or sequence across a population. So we'll look at 3 trials where this same sequence exists. And here the color code is just indicating the neuron ID that fired the spike. When each neuron spikes only one time, like this example, you can do the nice vector representation of your spike pattern. And from there, they're really easy to work with mathematically and compare your trials. But the moment that you introduce an extra spike, so that second one, make this work. Okay, that here, the second neuron has that second spike. That second spike doesn't actually fit into your vector. And it's unclear how you represent that neuron's activity. So do you represent it with the first spike, the second one? Do you average them in some way? And then more than that, if you want to allow for different numbers of spikes across neurons and across trials as what actually happens in real data, the question becomes how you compare these more complicated patterns when there's no longer a really clear and convenient mathematical representation for them. And notably, even in this really simple example, you couldn't actually recover the sequences by looking at the first spikes or the spike bursts. So what we need is a method that can characterize the complete set of spikes and still find that structure if it's behaviorally relevant without making any simplifications or assumptions ahead of time about which spikes might be involved in that structure. So to address that challenge of characterizing the complete set of spikes, we've derived a transform that takes a pattern of spikes across a population of neurons, and it decomposes it into a set of simple and overlapping spike sequences, which we call helices, and which you can think about as building blocks of spike patterns. And you'll notice that the plot on the left, the spikes have changed color to match the helix that they best align with. Now, to make that decomposition work, we needed the spikes that line up with a helix to sum together in some way, but the spikes that don't line up with a helix need to cancel each other out. And that cancellation wasn't possible if we just consider the sequences playing out over time. So we had to adopt a different geometry where time gets wrapped around a circle, and then neurons are stacked vertically into a cylinder. So here in this cylindrical geometry, each circular slice corresponds to one neuron in the population, and then the spikes from those neurons get positioned around the circle according to their timing. I'll just note here that the geometry of this helix or this cylinder has no direct correspondence to the physical geometry of your recording. And the neuron ID and the way you stack them is actually arbitrary. It won't affect the link to behavior in the end. So I will get to those details later, but I just wanted to put a note there so that you didn't think the cylinder was directly tied to your recording device somehow. So in this geometry, these simple spike sequences that are our building blocks become helices that wrap around the cylinder with specific spacings and directions. So they're sort of a set of relationships in space and time between the spikes. And at a technical level, you can think about them as a Fourier basis in a cylindrical geometry, which we can use to deconstruct these arbitrary spike patterns. Now, we do that decomposition using a generalization of the inner product operation. And I'll go into the details of that in the next section, but the main point is that we developed that operation to handle comparisons between spike trains that have different numbers of spikes. So we're comparing a arbitrarily sized spike pattern to a helix that always has one spike per neuron. Yes.
Question. Do you have to order the neurons by like to something, or like you don't have to do any like sort of?
So I usually just in practice, sort of whatever the ordering is that comes off the array is fine, but you can actually scramble that as long as it's the same ordering across all of your trials. It ultimately doesn't matter. It will affect the output of the transform, but it will not affect any clustering or the relationships between the spike patterns that you get out. So it's like an isometry in that way. And we can talk about the details of that later once we get a bit further. Yes. Yeah. So that's what the generalized inner product makes that comparison. And so like after we finish the big picture, I'll show you like all of the mechanics on how that one works. Sweet.
They have the same interval between two spikes, or it's just like
in the helices, they do. So in those like building blocks, they are all evenly spaced out between them. Like in a Fourier basis, how you would have the different waves and the different wave numbers, but by combining them and like using them as sort of these building blocks where you decompose and you say how the strength to which one is present and its timing, you can construct arbitrary patterns from them. So this decomposition, it gives us a mathematically rigorous way to characterize the structure of these arbitrary spike patterns. The output of the transform describes the contribution of each of the building block helices to your spike pattern in question. So kind of like Fourier analysis, that contribution is a complex number. And as I was just mentioning, it has an amplitude that represents the strength of how much a helix is present in your data, and a phase that represents its timing. So here in this plot, each dot is the contribution of one helix, and then the colored dots correspond to the helices that I've depicted in the center. And you'll notice I picked them because they have higher amplitudes than the gray dots, so they're farther away from the center of that plot, which means there were more spikes that aligned with them that I could change the color in on the plot on the left. So in the end, once you do this transform, this produces a vector, which describes the spike pattern in terms of a set of simple building blocks. So here in this vector, now each component corresponds to one of the dots in the previous plot. So each component of the vector represents the extent to which a specific building block is present in your spike pattern, a particular helix. So the key difference from other approaches is this vector does not just, so each component doesn't just represent the spikes of one neuron, but it's actually representing a sub pattern and how much that's present. And we refer to this output of the transform as the spike pattern's fingerprint because it provides a unique description of how you could build that spike pattern from a set of simple building blocks. So now that you have the big picture, we can take a look at the math that powers that decomposition.
You go from the vector back to the helices.
So we're working on that part. So so we have it's unique up to a very specific transformation where like a very specific, every neuron spikes need to be rotated in a certain way to have two spike patterns that end up at the same fingerprint, which has never actually happened in the actual data. And then I'll show you some examples of how we can look at the, like in the data applications, we can look at the that vector and then go back to the spikes on an individual trial that correspond to one of those helices. So that's sort of going backwards from this, yeah. Does that answer your question? Okay, cool. We can get to it more later. Yeah.
Just make one comment. In the human brain, all neurons do not spike. So for example, in the retina, which is very thin, the photoreceptors and the second order neurons make graded potentials. And it's only the ganglion cells whose axons leave in the optic nerve that have spikes.
Mm-hmm.
And crystals.
In this, yeah, okay, thank you.
Okay. But there were, in terms of long distance, yeah, it's the ganglion cells.
Right.
But you know, in the cortex, which is where most of the data comes from, it's by you know, wide margin, except for the astrocytes, you're gonna get spikes from the other things.
It's a really great point to bring that one up because I think like the way you would need to analyze that is really different. And so yeah.
Just.
But yeah, no, it's super important. Yeah. So to go into the details of the generalized inner product and how we do this decomposition, imagine you have a pattern of M spikes that are fired by N neurons over some amount of time t. And we're going to start by comparing this more complicated spike pattern to our set of simple elements called helices. And as I said before, those are the simple spike sequences that have different spatial frequencies and different directions across the population. So to make that comparison, we write out our spike pattern as two vectors, where the jth element denotes the jth spike. It has a neuron ID we'll call yj and a corresponding time xj. Now, our method uses complex valued spike times. So in that cylindrical geometry that I was telling you about, each circular slice is given by the unit circle in the complex plane. And so we need to transform the recorded time tj into the complex plane by first normalizing the spike times within your window of analysis. That window could be an experimental trial, an onset transient, a cycle of a population rhythm, a certain behavior, sort of whatever is relevant for you to be analyzing. And once you've selected that window, you normalize the spike times, and then you linearly map to a phase value, theta j, and exponentiate to obtain a number xj, which is on the unit circle in the complex plane. And I told you before, in the cylindrical geometry, where each circular slice corresponds to one neuron, we position the spikes of that neuron around the circle according to its timing. This transformation is how we do that. So now if we look at the helix, we'll call it helix K because it's the kth element in the set of helices we're going to use for the decomposition. H sub n indexes the nth element of that helix, and it is a complex valued number as well. It lives on the unit circle in the complex plane, just like our spikes. And we're going to use the dot product to measure the alignment between the spikes and the helix elements. So that results in another complex valued number. It also lives on the unit circle, and it represents the phase shift between the spike and the helix element. So now that you're oriented to our notation, we'll just walk through a really simple example of how that comparison works in practice. So we're going to compute the contribution of this basis helix to the simple spike pattern that's depicted here on the right. Now, most decomposition methods rely on the standard inner product to do that computation. The standard inner product moves element-wise along two vectors, always comparing elements at the same position in both vectors, which means that the standard inner product would require two input vectors to be the same size. And the problem with that in our case is that we want to be able to study spike patterns of any size where every neuron can spike as many times as it wants to or not at all. And so the generalized inner product changes that operation by introducing an indexing operation where we use the neuron IDs that are attached to each spike to dictate which element of the helix you should be comparing to. So let me demonstrate what I mean by that. So we'll start with the first spike. We look at the ID of the neuron, and we use that to index into the helix to know what element of that helix we should compare the spike to. We then take the dot product between the spike and the element of the helix specified by the neuron ID to get a complex valued number that measures the alignment between the first spike in our pattern and that helix. Then we move to the second spike. We use the neuron ID to index into the helix. We measure the alignment using the dot product, and we keep track of the result. And we just repeat that process, moving spike by spike through the spike pattern, always comparing the spike times to the element of our helix that's specified by the neuron IDs. Yes.
These electrodes, they consider one signal, or they have many signals in one electrode?
So that depends on your spike sorting, I guess. And if you're doing multi-unit, if you're looking at multi-unit activity, if you're looking at like well-isolated single units from the electrodes. But in this case, I would be like thinking a single neuron, potentially a multi-unit, but I haven't actually tried it in a multi-unit activity yet. Mhm.
Each electrode one signal.
Yes, but you could have many independently isolated units on each electrode, just by spike sorting.
Yeah, because you know that you have two parameters important there, the distance and the time. And then I don't know that in this case, how you consider the distance and between the electrodes, or where you get the sense, the signal.
These are, so this is, this method is really just starting with like a single unit isolated on the spikes of the single unit isolated on an electrode. So for example, you've already, the experimental group, for example, will do spike sorting, and now they have the spikes isolated from on these, for example, on the Neuropixel array, they may have 200 well isolated single units simultaneously recorded.
So I have a question. So
Yeah.
there's an assumption here that you have fewer spatial coordinates and time could be infinite, right? So you're kind of trading off the complexity and an infinite time to space that's very finite. But what if, we get better resolution in electrodes and there's more spatial times? And maybe we just want to look at a millisecond, and now space grows up, right? So is this method commutative? Can you like switch?
Can you switch it around? Oh, that's a really interesting question. Like, could you have either a spike or not a spike, but then over space?
Yes.
I haven't thought about that before. That's a great suggestion, though, to like actually explore a little bit more. It's really interesting. Yeah. So once you've done this sort of indexing operation, at the end, like in the standard inner product, you then sum up all of these points to get a value, mu k, which summarizes the contribution of that helix to the spike pattern. So this is the same mu k that we talked about before that has an amplitude that represents the strength of the contribution and a phase that represents its timing. And to build some intuition for what that value means, suppose we're going to compute the contribution of this helix to two different spike patterns. The first one contains a close replica of the helix here in green, but that's not the case for the second spike pattern. So when we compute the contribution of that helix to spike pattern 1, the spikes that line up closely with that helix result in points that are close to the positive real axis because the phase shifts between those spikes and the helix elements are very small. And when we do the same thing for the second spike pattern, we get a collection of points that's more evenly spread out around the unit circle. Now, each of those points is the end of a unit vector. Our next step is to add them up. And when we compute that sum for the second spike pattern, the points cancel each other out, resulting in some complex number that has a small magnitude and some arbitrary angle. But in spike pattern 1, those gray points are still working to cancel each other out, but the green points that are all headed in the same direction add together so that mu k is far from the unit circle. Now, the magnitude of mu k is related to the number of spikes that are lining up with the helix, and the argument specifies what phase shift is required to achieve that alignment. In practice, we divide it by the number of neurons so that if you had a perfect copy of that helix hiding in your spike pattern, mu k would have length 1. And then in our data applications, we use randomly generated spike patterns with the same number of neurons and spikes that help us separate between these two cases when the magnitude is small versus when it's large. So we can start to look for when there's more structure present than what you would expect by chance if you just randomly generated spike patterns. So computing the contribution for a single helix, as in the previous example, is useful for detecting structured subpatterns like sequences that could be present in the spiking data. But by extending that to include a large set of different helices, it's possible to fully decompose your spike pattern to obtain a complex valued vector, mu, which fully describes the fine scale structure in your spike pattern S, using this set of helices, which we'll call H. And that decomposition is captured in an operator psi. Again, we're going to refer to the output of this operator as the spike pattern's fingerprint. All right, so to summarize, the MSDHT, or the multisample discrete helix transform, is given by this operator psi, which is defined for a set of helices, H. It takes as input a spike pattern S, which is defined in the cylindrical geometry, and it outputs a complex valued vector mu. And each component of that vector is given by the generalized inner product between the spike pattern and the helix. Each component describes the contribution of that helix to the spike pattern S, and we compute it as a normalized sum of phase offsets between spikes in the spike pattern and spikes in the helix. So here Yj is the ID of the neuron that fired spike J, and we're using that to index into the helix. So we're comparing the time of the spike xj in the spike pattern to the spike from the same neuron in the helix. And in that way, if any of that neuron's spikes line up with the helix, this operation can detect that. It doesn't matter if it's the first spike or the last spike or some spike in the middle. And so now with this transform, we can assign each spike pattern to a fingerprint, which is just a vector in Cn, where there are already well-defined distance measures that we can use to compare the spike patterns. So the next thing we'll look at is an example of how that comparison works in practice. Yeah. Yeah, it's a great question. So we, the like sort of standard way that I've been doing it, that seems to be working nicely in the experimental data, is using sort of the like full basis, using that word a little bit loosely, of the number of helices that correspond to the number of neurons. So you essentially have these increasing wave numbers that then flip direction and go back down in the other way. And when you're looking at a recording of a couple hundred cells, for example, that seems to be a really good amount to get good sort of dimensionality reduction, as I'll show you after. When we're starting to apply it to the like large scale spiking networks that we have, when there's thousands and thousands of cells, I'll look at like a truncated version of that with 100 or so on sort of each side, and that seems to work well as well. So they're orthogonal, but you don't necessarily need like the full.
What's X and Y here?
X is the spike time, and Y is the neuron ID that corresponds to that spike. Yeah.
It's incorporating time warping. It seems that due to external delays and synaptic transmission and noise, sometimes you get the same sequence, but just more spread or some jitter in spike times, and it kind of makes sense. And you also, I think, there's literature about that to include some time warping.
Yeah, that's a great question. And that's actually one of the benefits of doing the wrapping of time around the circle, because that kind of naturally can account for some of those shifts and rotations using just like the symmetry and the complex plane, instead of needing a algorithm to do that time warping for you. I've seen some other ones that sort of have like a template matching type thing, but stretched and compressed in time. Exactly. So here, the sort of the price that you would pay is if you have more jitter in your spike times, then those green points that were close to the positive real axis will get a little more spread out. And so across your trials, you might get a little more jitter in the value of mu k if you're trying to detect a repeated pattern. But it seems so far to handle that case like pretty well. Yeah.
And so if one were to assume that the helix space is complete, right? Could you, and you the fingerprint, could you treat this as a sparse coding problem and try to figure out what spikes happened in the past? Is that something fair direction?
It's a great question. And we've actually started looking at if you had a fingerprint, could you just go back to an arbitrary spike pattern that would get you that fingerprint? And we're sort of doing that in an algorithmic fashion right now, because if you didn't know the number of spikes you had, like it becomes a sort of wildly unconstrained problem if you have like the solution to n equations, which can have m variables and you don't actually know what m is, and both of those have but like algorithmically you can sort of make that work if you maybe set some constraints, or if you, in the case where you knew how many times each neuron spiked, then you could go backwards. So there's still some things that we're working on there, but it's a great question. It's super interesting, I think.
I have a quick question online, if I may.
Number of helices and number of neurons.
Yes.
So like we don't consider like the rotation, like neuronal rotation, like we like kind of like shuffle the index of the helix.
So a little bit. So the helices will always follow the same pattern where they're sort of increasing their spatial frequency. You can change the neuron IDs, but you have to do that across all trials. It needs to be consistent. Otherwise, you're not comparing like apples to apples anymore. If a neuron is 1 in spike pattern 1, and then all of a sudden it's 3 somewhere else.
Yeah, I think it's because like since the result is also like an n-dimensional vector. So that like reminds me of let's say we just like average the like number of spikes across this time. So we got this like response vector. This is also like an n-dimensional.
Yes.
Vector. So I wonder, like, let's say for the later case, the time information is locked. I wonder what information is locked here.
That's also a great question, and something we've been playing around with is looking at sort of using this method and like the dimensionality reduction bits that follow from this n-dimensional vector to compare like what happens if you get rid of all your timing information and each component represents the count of a neuron compared to this one. It's kind of interesting. So the helices in the sense are sort of like an over complete basis where there's relationships between them, and sort of if you know the contribution of a certain spike to one helix, you sort of can know what it's going to do to all the other helices and work it out that way. But it's a little different just because the each component is representing a whole sub pattern, not a single neuron. So there's still some things we're like, yeah, working out there.
So the control would be just to give a bunch of Poisson spike trains independent.
Mm-hmm.
And how would that look on your helices?
That's a great question, and it's a good thing that we should do. Lyle, you look like you want to jump in there.
So we can tag the helix by shuffling the spike times and then comparing to random.
No, I'd like to see if you shuffle something that already has structure, but I want to start with something that has no structure. In other words, you get false structure reports.
So you,
from random numbers, random spikes.
I understand the question better. Yeah, so you would end up with very small magnitudes for your helices. So like super, super small compared to the number of neurons. And so that wouldn't be like counted as a detection because we're always comparing to that control. So where we're randomly generating a set of spikes, same number of neurons, same number of spikes, whatever timing. And then we only count a helix as being detected if the magnitude of it exceeds what you would expect by doing that 1,000 times or something.
Oh, you have that.
So that's part of the control for not structure. Yes. Yeah.
But also the Poisson constraint allows you to know the number of events that are going to happen, and then you can try to recover the times. Because when I ask a question, you know basically you know the number of events that are going to happen.
Mm-hmm.
And if you specify a Poisson, then you know the expected number of, right? So then you could try to
Right,
recover.
and then you can work backwards. Yeah, and we've started developing it that not in equations yet, but in just like an algorithmic implementation where you sort of throw some spikes down and then ask, did that get me closer to the fingerprint or farther, and sort of start adjusting them that way. We're still working on that part, but sort of headed in that direction. And it would be cool if you had that constraint to be able to actually get it in an equation. Yeah.
But it's important to speak right out of proof to show that the transform is unique.
Yep.
Very kind of minor degenerate cases. So
Yep.
we can so see that this transform is unique and has that property, and so we know that we're going to these unique points and see it.
Yeah.
So that's an important property related to the invertibility.
Yeah.
Sorry, another question.
Yeah.
So like, if I understand like part about this like helix like space is kind of complete, but it's not like intuitive enough to me. So I wonder in which sense it is complete. Because like for this like neural matrix, it has n by t figure freedom. So if you are only having n like items here, so in which sense it is complete? Like I mean, like is this like descriptive enough for a lot of cases?
Right, I see what you're saying. Yes, so with the uniqueness part, what we're finding is that you can uniquely describe essentially any spike pattern that you put into it using them. The case where that would maybe break down or not be complete enough would be the case Lyle was just sort of talking about, where the spikes of each neuron, if they are all in one spike pattern, if all of the spikes are there and just rotated to be perfectly symmetric, but every neuron had to have that same rotation, then you would end up at the same point in CN. But that is the only case where that would happen. And so far, I have not found that in experimental data. Yeah. Cool. So to show you how we can start using this distance in CN to compare the spike patterns, this is just a constructed example where I'm generating 3 different types of trials. And I do that by embedding a partial copy of 1 or 2 helices into just a bunch of random spikes. So in this example, trial 1 and 10 come from the same set of helices. So they're like indicated in purple. And we can see just by eye, there's a couple similar sort of diagonal lines of spiking in there. I don't think you can see my mouse, but the other trial in green comes from a different set of helices. And the idea is that this method should be able to pull out 3 different clusters of trials corresponding to 3 different types of spike patterns. Yes.
What do you mean different set of helices? You mean, don't they have to be the same basis?
Yes, but so what I did is I sort of overrepresented one of them in the spike pattern. So I put random spikes, and then I took part of one helix, say helix 2, and said, okay, all of the purple trials will have half of helix 2 hidden in them, and all of the green trials will have helix 8. And then in theory, we should be able to recover those clusters of trials. This is just sort of like a demo for how that would work. First step is to compute the fingerprints for all of those trials. So here I just have 25 trials of each. And even by eye, looking at these helix amplitudes, you can tell that they are separate. So in this plot, each column is the fingerprint for one of the trials, and we're looking at the amplitudes. And you can see these sort of high-valued horizontal bands across some of the trials, which correspond to the helices that I used to generate those trials. So just by eye, you can see we should be able to get the 3 clusters out. From here, what we do is we compute the distance in CN between all of the pairs of the spike patterns.
CN?
Complex valued numbers, but with n dimensions. So it's like a vector where each component is complex. So the just like the natural sort of distance between these fingerprints. And what you can see is that on the diagonal, we have these low valued blocks, which means that the spike patterns that were created in a similar way that came from the same group are all being sent to points that are close together in the Euclidean sense. And from here, we can use dimensionality reduction. So we use spectral graph clustering for this to produce a projection where each dot corresponds to one spike pattern, and they get positioned in this like similarity space with their distances in this space related to the distances between the spike patterns in CN. So here, these dots, they're colored by this type of spike pattern that happened on that trial. And so you can see that these clusters are really clearly separable in this like simple generated case, but just to sort of show you how the process will work as we move into the experimental applications. And then I'll just note that the cell relabeling, and we can go into more detail later too, but the cell relabeling, if you scramble your neurons, will affect the first plot. So the blue plot, the output of the MSDHT, that's going to change, but it's an isometry. And so the distances between the spike patterns, so the second and the third plots, will not be affected by that relabeling. And that's the part where you start making links to behavior in your task, because like maybe looking at different conditions in your task. So the ordering of the neuron IDs doesn't matter to your ultimate link to behavior in the end. It will affect the output of the transform, but not the clustering. Cool. All right, so to get to some examples of our initial applications, as a first demonstration, we turned to neuropixel recordings from macaque area M1 during a motor task. And this is in experiments done by Jonathan Michaels and Andrew Przynski, also at Western. In this task, a robotic exoskeleton applies perturbations to the subject's arm in one of two directions. So we're starting here with a really simple example where they're in a kin arm, so they're in this like robot, and the robot will bump them either in an extension or a flexion, and their job is to return back to the center. It's a really large and salient perturbation that causes either an extension or flexion. And in the 100 milliseconds following that perturbation, the firing rates, if you average them across the population, they'll increase transiently. And in what follows, we are going to decompose and analyze the spike patterns that happen during that 100 millisecond response. So what we found was that some helices were detected more frequently on trials of a specific perturbation. So in this session, helix 2 was detected more often on the extension perturbation trials. So to show you this, I'm going to play you a movie where we plot the spikes across the extension trials that line up with this helix. It's going to be a subset of the spikes that I'm plotting, not every spike, just the ones that are lining up closely. So we start by plotting the spikes on one trial, then a second trial, a third trial, and we'll keep overlaying all of the extension trials. And as I do that, I will gradually decrease the opacity so that we can see how much they're overlapping. And what we can see is this pattern is repeating reliably across trials. That's in the dark regions there, with some variability in timing from trial to trial, and that's the sort of smearing along the time axis. But what's interesting is if we do this exact same thing and generate the exact same plot, but for the other type of perturbation, so now the flexion trials, we don't actually see any of that structure. What we do see on the flexion trials is a different helix that's reliably detected, which is helix 8. And on those trials, again, same plot, so we're overlaying subsets of spikes that line up with a helix that was detected frequently. Now, if we look at helix 8 and we return to the blue extension trials, we don't actually find helix 8 there. So together, this is showing that our transform can discover robustly repeating and behaviorally relevant structure in the spike patterns that are happening in this task. And then further, if we take any individual trial and we just ask, did we see helix 2 or did we see helix 8, we can guess what perturbation happened with 94% accuracy.
There was only one helix per deflection, type of deflection.
There were many. So
There are many.
there are many perturbation-specific helices that happen in M1 in this area. What's really interesting, they had other recordings from other areas. And so in this 100 milliseconds, we couldn't find any helices that cared about the perturbation in PFC, for example, which kind of makes sense because in that 100 milliseconds, PFC shouldn't yet know about that perturbation. But these are just examples, and I picked the lower spatial frequency ones to show you because it's a little easier to see in the plots. But to show you how that decoding works, so we know that helix 2 happens on the extension perturbations, helix 8 happens on flexion trials, so we can basically use these as templates to ask on any individual trial which spikes line up best. So on this example trial, we can see there's a number of blue spikes that are lining up closely with our blue template, but the pink spikes are kind of all over the place. And the blue spikes are the ones that contributed to helix 2 on this trial, and the pink spikes contributed to helix 8 on that trial. And so here we would correctly guess that it's an extension trial because the blue spikes lined up better. The same thing happens on this trial. So more blue spikes line up, pink spikes don't really line up, and on this trial. But on this other trial, for example, we can see the blue spikes don't line up with our template of what we would expect to see from helix 2 on an extension trial. And we actually have a lot of the pink spikes that are lining up closely with our pink flexion template. So here we would correctly guess it's a flexion trial. And then again, something similar happens here where there are a few blue spikes that line up, but not very many. There are many pink spikes that line up and here. And so when we think about that from the perspective of the helix transform, the decoder just needs to look at the contribution of helix 2 and helix 8, so mu2 and mu8, and try to separate those vectors of length 2. And you can see here, when we do that, we get pretty good clustering using those 2 helices only. But if you include more helices, and that means more spikes and more neurons reliably across trials, we can really start to pull those clusters apart and drive up our decoding accuracy. So, yeah.
What makes a spike blue or orange?
Yeah, so great question. So we do the transform and we get this vector that tells us the magnitude and the timing of each helix in our data. And we call a helix that it's detected if it has a high magnitude. So there's a lot of spikes that line up with it. Then I look at what is the timing of that helix. So what's the argument of that component in the vector? And you can go back to your spike pattern to ask which of the spikes are lined up with a helix and that phase shift. So those are the spikes that sort of were the ones that grouped together in like that circle plot for the generalized inner product, and we're driving that helix contribution to be far away from the unit circle. So we can, go backwards from the transform to the spikes on a single trial for which spikes caused each part of the
So
transform.
can you see it without your eye?
No. Yeah, so
They're kind of hidden.
mhm,
Patterns are hidden.
yes.
How many units do you think you would need to see like one helix with like a big enough magnitude to consider it, you know?
It's related to the fraction of neurons. So it is related to the size of the of the population you're considering and the number of spikes that are happening. So depending on the length of time that you care about and like your firing rates, that will change what we count as sort of our control for how large the magnitude should be. So in this example, there are very few spikes that are happening in general across this population in 100 milliseconds. So you need fewer neurons to say that it's more structure than you would expect by chance. If you went to like a second long recording, you would then need more spikes to call that significant.
So this is M1.
Yes,
So we know that there is a rate change too, right?
yes, exactly.
How is the rate change related to this, helix code?
That's a great question. So exactly from the rate you would expect these to be separable from the firing rates. Some neurons are going up, some neurons are going down. What we're trying to do is look at a sort of a different aspect of that right now to see if there's these timing, but the neurons that participate in a helix may have a specific rate change as well. And that's something that we haven't yet like gone into the single neuron, but something that's really interesting is like which neurons are participating and like what are they doing in terms of the other types of coding that could be happening. So I don't have an answer yet, but
But there's also presumably the perturbation moves the arm over some 100 milliseconds or something. So you might be able to pick out some change in the helices or something, if you broke that into say 10 time intervals. And is that something that you could do too?
that's a great, so looking at like the fingerprint and how
Yeah,
it changes
the
from
fingerprint
time to
changes
time. Yeah, that's something that's been on our list and what we've looked at a little bit, not in this data set, but yeah, it's a great point. And you could trace like trajectories through CN even about the way that the spike patterns are changing.
And so could you say something about more of a semantic model, like separation, flexion, extension seems to occur on a linear axis, so you could say it's a spatial representation. But if you have a semantic task where you don't really move along an axis, say you clap versus you jump, those two are not carried on an axis. But
Yes.
could you still kind of separate?
Yeah.
semantic from interaction?
So that's actually leads in really nicely to the next application that we have, which is a bit more of a naturalistic task. It's not quite that like different semantic motor thing, but it is more complex behavior that plays out over more time and not just in this constrained axis. So this example is dual Utah array recordings that are from macaque PFC during a working memory task that,
I have a different question.
Yes.
So over sequence of days, the monkey's brain moves and the electrodes move and you'll get different neurons.
yes.
Will the representation you get out when you sample from different neurons give you the same helices?
So every session we treat separately. So we wouldn't combine multiple sessions and suggest that the same neurons would have the same IDs. So you might end up with, you'll likely end up with like a different helix. So it's not specifically that helix 2 and like that number 2 is what matters here. It's more the relationships between the neurons and their timing that are part of that. And that can change across days. So we wouldn't combine it.
You can't compare it between.
Not unless you knew that you had like stable neuron recordings across days, I think. You could do other things like looking at, do we have similar numbers of
But
helices that
presumably if
care?
You have two arrays recording from different neurons and did the same analysis, you should see something similar in terms of the helices, right? For all the trials, same trials, right? In other words, it's a distributed representation that's shared by the neurons.
Oh, I see what you're saying. Yes. Yeah, that one makes sense. I think it's just more the across
And in
days.
fact, you
Yeah.
could probably just split your data set in half, analyze them separately and show that, there's something that's conserved across the two populations.
That's a,
No, this is at the same time. It's not overtime. No, probably there's real problem with drifting representations, right? So if you just wait a week and you have the same neurons exactly, you may not get the same results.
mhm.
But one of the controls is to split the recording session in half, and you find similar helices if you did that.
Yes, not the population. So I like not splitting half neuron, like half of the neurons and half of the neurons, but if you split even and odd trials, for example, you get the same thing out. So it doesn't matter if you like subsample your trials, you would end up with the same helices. If you change the population of neurons, as I said, it will affect the output of the transform. Ultimately, it won't affect later on your clustering, but like the numbers of the helices that come out will change. Sweet.
Number of the helices, but the helices.
So like the helix 2 versus helix 8, like the name of the helix that comes out as being the detected one, if you scramble your neurons, that part will change. But the fact that all extension trials, for example, had that same helix shared, that part won't change. Cool. Okay, so to go back to the
It's a permutation, right?
yeah,
What
yeah.
you're saying is permutation.
Exactly. Yeah. If you permute and then what it leads to is it's an isometry in that the magnitude of your fingerprint, like the magnitude of the whole vector is the same across your permutations, but each independent component will be different. Cool. Okay. So the second example we have, oh, actually we are running low on time too. So you think so? Okay. Okay, cool. This is the last one. So this is dual Utah array recordings. It's from Macaque PFC and it's a working memory task, takes place in a virtual environment. So you can see that on the top, it's this task setup and below it's the virtual environment. There are 9 target locations, which are all these red bars here that are organized into 3 rows and 3 columns. Depicted here, the columns from above. So by columns, I mean the depth dimension. That part is important. As you'll see later. And I'll just start by playing you a video of the task. So you can see the 3 epochs of the task, and you'll also see this white moving circle, which represents the subject's eye position. So during the Q period, a target location is presented. The animal can look around freely, but they can't move. During the delay, that target disappears. And then during the response epoch, the movement turns on and they use a joystick to navigate to the remembered location to receive a reward. So in previous work, we found that sequential activity encoded working memory content during this task. So we made that big simplification I was talking about at the start, where we simplified to a single spiking event per cell, we sorted the neurons, and we obtained these sequences. These sequences were made up of brief elevations in firing rate, which I'm depicting here as in the black, where the neurons would jump up to their peak firing rates for 100 or 200 milliseconds, and then they would drop back down to baseline. And what we found was that the ordering and the timing of those sequences was similar on trials of the same target, and especially trials in the same column. So you can see that column clustering here, where we're projecting into that similarity space I showed you before with the clusters. And we're looking at the centroids of each target condition. So this is like the average sequence for each of the targets. And the thing that we're interested in is the fact that all of those blue dots that are corresponding to the left column are close together, and they're far separate from the yellow dots, which correspond to the right column, and the green dots, which are the center column. Now,
It's 3 sections.
exactly, yeah. So like when you're looking out into the circular arena, sort of the 3 targets on the left.
But
So one target will be presented, and by the column, I mean like the location of the target that's presented, and then that one is present for the whole task. So they see it during the queue, it disappears, they remember it during the delay, and then they have to navigate to it. And this will probably make it a little more clear why I'm separating by columns. So these are example paths that they would take to each of the targets. And you'll notice that they sort of, on these correct trials, they have this sort of stereotyped, they move in a similar way to the 3 targets that are all in one column. And in spatial column, exactly. Yeah. And so in our previous work, we were relating the sequences to these trajectories that they were taking through the VR and in that way. But now that we have the MSDHT, we can go to these more complicated patterns of the peak firing events, because the neurons weren't just elevating their firing rate once per trial, they were actually firing many times per trial. So here the neuron IDs aren't sorted, and the MSDHT allows us to handle this more general pattern of spiking activity across the population. So what I'm going to show you today is that we can confirm the previous results. So we still find that the spike patterns are clustering largely by column. Here, for example, we're looking at the centroids. We still see these sort of 3 groups of them, but now we can really go to the individual trials and start looking for patterns there. So here I've colored them by the column now, because that's sort of the grouping that we care about. And what's really interesting is we can use these spike patterns to predict the specific choices and the errors that they make on individual trials. So we can really tie the spike patterns directly to the navigation paths in a more concrete way. And what I'm going to show you is related to that little blue dot that's sitting in the yellow cluster there in the spike patterns. Yes.
How changed with the learning process? Because this one you have to learn, right? And then how changed it?
It's a great question. And we didn't have the recordings while they were learning the task. So they were trained on the task first, and then they started doing the recording, but I would love to know what happens. Yeah.
What would happen to the blue sheet?
The dot?
Yeah.
I will show you. So the transform is picking up the structure in the spike patterns in the PFC. That's giving us insight into specific choices that they're making at a single trial level, whether that's navigating wrong through the environment and making a mistake and getting the trial wrong, or whether they actually get the trial right, but doing it in a way that breaks from their stereotypical behavior. So in this plot, what we're looking at is the similarity space for spike patterns at the start of navigation. Here, each dot corresponds to 1 trial, and they're colored by the column on that trial, like in the little legend there. I'll also just note this projection is produced using all trials, so correct and incorrect. I've just split them up. We're only looking at correct for right now. And you can see there's clear clusters here. So the left column trials in blue are separated from the center in green, and the blue in and the right column in yellow. Too many colors and things, but the highlighted regions there, the shaded regions are just schematics to draw your eye to what I mean by those sort of 3 clusters. But you'll notice that there's also some of the left column trials that are clearly clustering with the right trials, like this one that I've circled in black here. And when we take a look at the navigation strategies on these left and the right trials, we can see there's a pretty clear separation between what the subjects would usually do on a blue versus a yellow trial. So what about that blue sheep, the blue spike pattern that's sitting in the yellow cluster? That trial is here. So this blue spike pattern, it looked more like a yellow spike pattern, and it turns out that the subject actually navigated more like a yellow trial. You can see they're starting as if it is a yellow trial. They get to the end, they didn't get a reward, they turned around and they fixed their mistake. So the spike pattern in PFC at the beginning of navigation can explain that choice. And then we can go to the incorrect trials. So again, this is on the same axis, and I'm using that same schematic to guide your eye to where the correct trial clusters are. Again, we can find examples of trials that are placed with the cluster of the wrong color. So for example, this one is again a blue trial, but this time it's an incorrect trial, and it's placed with the green correct column trials. Now, again, we can look at the navigation strategies that are typical for blue and green trials, and we can see what happened on this trial. So here, we can see how the position of the spike pattern relative to the other spike patterns can explain this specific error, where they navigated as though it were a green trial, and they never actually managed to fix that mistake and go to the correct trial, which is on the left, circled in the dark black. Now, these are just, yeah. Yes.
Looking at 1 and 2, but we moved to 2 and 3.
Yeah, so before we needed more to get good clustering. So there is a little bit of change in dimension 1, but it's less relevant. Like here, just the 2 axes is enough to get clustering. So I was just plotting it that way because it's easier to see. Yeah, so they are. It's like it's the spectral clustering thing, but it's not always the case that like a dimension with more variance is necessarily the most important one either. So the first if you look at the first dimension as well and have it in a 3D plot, you still see 3 nice clusters, but there's not too much going on in terms of the columns in that first dimension. So I'm just not plotting it here.
Memorizing the stuff.
I did not do the recordings for this task. I know that it was a long effort before I came into the lab, but yeah. Okay, so I've just showed you 2 examples.
Total that you use for this.
This one was between 200 and 300. So they had 2 96 channel Utah arrays. They did the spike sorting. So depending on the session, it was sort of in
That's
there.
actually a fairly small number.
Yeah,
Small fraction.
especially compared to now when you can have multiple neuropixels and stuff too. Yeah.
But that's amazing that you could. Just, in other words, you don't have to record many neurons in order to be able to know what the rat did, right? What the monkey did. Sorry. Right. In other words, which means there's a lot of redundant information throughout the entire PFC. Right.
Right, and like a lot being represented because a lot of things can also be decoded.
That's a very low dimensional benefit.
Or a simple task.
Well, for a monkey, you have to train them for months to get there to do this, right? It doesn't come, you just can't tell the monkey, go to the right.
Well, yeah, but is
You
that
see it
because
right-
it's outside of is that because it's too complex for them or because it's too far outside of their ecological niche?
Because they don't talk to.
This is a difficult task, right? You have to train a monkey for a long time, but.
Yeah.
This task, we were able to compare virtual reality and the simple ocular motor response or ODR task, right? The simplest working memory task. And what we found is that the sequences really specifically emerge in the VR. So
Yes.
more complex task and simpler neural response, which was a surprising part of this. And that was part of the first paper before we had this the new transfer. But it does seem like a more complex and naturalistic task actually gave rise to a simpler neuronal environment.
Sweet. And that actually is a great way to wrap up. I was just gonna say, I only showed you 2 example trials, but that pattern actually holds across all of the trials and sessions in that the path that they take on a trial is significantly more similar to whichever is the closest cluster of spike patterns rather than the correctly colored cluster of spike patterns. So it's an example of how you could use this transform in the context of these more naturalistic behaviors to start looking at structure and spike patterns at a single trial level and how that can tell you about the subject making a mistake and navigating the wrong direction in VR.
But in this picture, in the incorrect trials, you have blue dots in the blue cluster.
Yep.
So
those would be,
happen?
yeah, no, that's a great point. So those would be ones where they took similar paths to a regular blue cluster, but they might not have gotten close. So the radius for a target was quite small for them to actually get the trial right. So there are examples like those ones where they did navigate the right way and they just kind of were a little bit outside of where they were supposed to be and they never quite got it. So there are ones like that as well.
So there would be like a crack going towards the blue circle that's missing.
Yes, like missing it around the outside or missing it slightly to the inside. Yeah.
This is based off the delay period, not the navigation.
This is the beginning of navigation. So the navigation plays out over up to 10 seconds. I'm looking at the first like 1 and a bit here. Yeah. Great. So just to conclude that I showed you a new mathematical transform, lets you characterize fine scale structure in large scale spike recordings with no restrictions on the number of spikes or neurons that you can include in your analysis. And I showed you some initial applications of how we can use it to detect structure and spike patterns from structure that plays out over 100 milliseconds in M1 to these longer timescale patterns in prefrontal cortex that tell us about the specific types of errors and choices on single trials. And then I just wanted to end by thanking all of our collaborators on the experimental and the theory sides of the project, my lab, especially Roberto, who's also here today, and Lyle, and all of our funding and you guys for listening. Thank you.
Didn't have as many helices as neurons. So there like some way you can say like some of these aren't really that present in this data, but some of these.
Yeah, that's a great question. So we'll use the randomly generated spike patterns to compare to ask whether it like each helix was significant or detected on a trial. And then you can start to do things like look at how many trials was a certain helix detected compared to another helix. And you will definitely like, depending on the application, I've found certain helices being present more and other ones less and that sort of thing. And just looking at sort of histograms of when they were detected or not is a helpful way that I've been doing it. That's an interesting idea, like in terms of how many of the spikes are actually contributing to a certain one. That comes from the magnitude of the helix itself. So you can look at what's the magnitude compared to other helices and rank them that way. That's actually really interesting. I haven't tried that, but it's a great suggestion. Yeah.
Why don't you choose like this way traveling with like helix bases instead of like something compared to like more traditional like 2D discrete Fourier bases? What is the advantage?
It's a great point. So the helices are representing a set of relationships in the spiking across space and time. And the cylinder essentially allows you, if you want a helix, let's see if I have a picture of them. So for example, this helix 2, if you shift it, say up one cell, for example, or down one cell, you might actually want that to be similar if it's like just sort of slightly offset in the population. And the cylinder with the wrapping lets you do that. I guess it wasn't necessarily the case that that would be relevant in the data. So that the question is then, is it, and are there experimental applications where this is making sense kind of? But yeah, there's definitely other geometries that you could explore and what we'll probably look at in the future, but for now it's working. Yeah.
So there's a, if you look at continuous signals, there's a related algorithm called a Hilbert transform, which allows you to pull out things like this. And I wonder if there's a continuous version of your helix. Continuous signals, like we were talking before.
That's an interesting idea because we've thought about it in terms of kind of like doing a Fourier transform if you had multiple samples per every time point, which isn't quite going to the continuous, but sort of like more towards the idea of a continuous, like continuously sampled.
This might be more interesting.
Yeah, it's a great point.
And like you can explore some dynamics in the system, right? Like, because different helix will represent sort of like different frequencies in the spatial like waves, something like that. That's why I'm asking this question, like if there are logical reasons or like maybe this helix is like we need much less number of helices to represent entire like spaces than compared to like Fourier basis. I can see that maybe like this helix transform basis is like captures the sort of like sparsity of the like bigger spaces. Something like that, which is quite interesting.
That is a really interesting point.
But it's still on the Fourier argument. So then, could you, for real-time fingerprint measurement, right? Could you bin time and repeat the process over time and kind of create a spectrogram to give a moving fingerprint, or you have to get an entire time and process offline? Because imagine you want to do control, right? You're constantly getting these fingerprints that tell you to move to correct whatever is going on. But it strikes me that you have to get all these spikes in post processing. So can you speak to like a real-time fingerprint measurement?
That's a great point. So we've actually been working on like writing out the equations for what happens when you shift your window a little bit, because there are clear relationships between the fingerprint in one window, and then if you slide it a bit, say to the next spike or to like 10 milliseconds in the future or something, where they're related. And so that transformation would sort of look smooth-ish through your like through CN if you're looking at the output of your fingerprint, and sort of studying that evolution over time is like something that's really interesting, what we're working on exploring now. Yeah. Mhm.
Sorry.
May I ask,
Okay, so there's this, measure of similarity between representations of representational distance matrices, like those.
Mhm.
And with those, you can compare two brains, or two species, et cetera. I'm wondering if, so there are relationships between relationships, and so I wonder if you did this on a different day and had different neurons, and did the same computation and then looked at the representations you get out and see if they have the same similarity structure to each other.
That's a really interesting idea, because that would sort of let you pull apart, like if helix 8 on one day is sort of playing like the same role as a different helix on the first recording session.
Right.
That's a good idea. I hadn't thought about that. That's really interesting.
Okay, so,
um,
the question is whether these helices have anything to do with behavior. In other words, how do you translate a helix into motor system? You have to decode it somehow, right? So do you have any ideas?
On decoding of the helix?
Decoding.
Right. So I would maybe think like there's work on how with like synaptic plasticity you can decode sequences. So maybe something like that would could decode helices and separate between them. Also something that we're looking at trying to implement, but haven't made it that far yet. Yeah. I don't know if Lyle has.
may I ask a question from overseas? Do you hear me? Okay, yeah. So I'm quite detached from all this, but this transform rings a bell. I seem to recall something called Huff transform. Does that have anything to do with your pyramids?
I'm not familiar with that one.
Okay, never mind.
No, there is. It's a simple version of what you've done, but not a helix. Just this histogram. It's a histogram of it.
I have a question as well. You can tell me if you can hear me.
Yes.
Oh, thank you. Okay, so spike patterns sometimes occur at different timescales. So Rafa Yusti had a while back a paper on these cortical motifs and songs. And I'm just curious if you can, if you're using a method, you can detect on the same bit of data patterns that occur at, say, 100 milliseconds time scale across a certain population, and then these patterns reoccurring as a pattern across like 2, 3, 4, 5 seconds. Or in other words, can you do patterns of patterns using the same approach? What would you do to do it, to achieve that?
That's a sorry for the echo. That's a really great question. I think there's a couple ways. So we've looked at, for example, extending the window to include more time, and then something that would be helix 2 in a shorter window would then become helix like half or whatever in the longer window, and you can sort of have relationships between them that way and use that to detect repeated patterns. But you could also window your time and ask if you get similar fingerprints or similar substructure that's being detected across those windows, and use that to say, well, helix 2 was repeating in all of these time bins.
But that would have to be hierarchical, right? We would have to have varying bins that kind of accumulate, just by keeping a single bin size will not show the hierarchy. You have to have multiple hierarchies of bins. Slide.
Oh, I see what you're saying. Like if one of them is, for example, 100 milliseconds and the other's like 120, it does depend on your windowing. So if you were to bin, say, 100 milliseconds and one of the patterns was slightly slower, so a few of the spikes kind of got dropped off the end when you made your window, you would, if the structure is sort of pretty robust, you would still end up detecting that even without a couple of this, with a few spikes missing, because you can add noise, you can delete spikes, and you can still detect a helix. But you might also look at more flexible windowing methods. So you might use like cycles of like a population rhythm, or like the broadband. If you look at like Lyle's generalized phase idea and use that to window your time, that could be sort of stretched and compressed a little bit. And you might find repeating patterns sort of within those naturally varying time windows, if that makes sense.
Well, we should probably thank the speaker again. Thank you.